Every single second, millions of times over, your body is pulling off this quiet, incredible trick. It's a process so fundamental to life, it is literally happening inside you right now. Today, we're going to dive into the cell cycle, the elegant, continuous story that every one of your cells lives out, from the moment it's born to the moment it divides to create new life.
So, what exactly is the cell cycle? Well, simply put, it's the entire life story of a somatic cell. And a somatic cell is basically any cell in your body that isn't for reproduction. You know, like a skin cell or a liver cell or a nerve cell. The cycle is the whole sequence of events it goes through from the moment it comes into being until it splits into two perfect, identical copies of itself. All right, to get our heads around this whole journey, we've broken it down into 5 main parts. We'll start with the big picture, then we'll dive deep into that long prep phase called interphase. We're going to put a special spotlight on the all-important DNA copying step, then cover the dramatic division in what's called the M phase, and finally, we'll meet the molecular guardians that make sure this whole process runs without a hitch.
Okay, so let's start with the 30,000-foot view. This whole incredibly organized dance can really be broken down into just 2 major phases.
Those 2 phases are interphase and the M phase. And here's the wild part. Interphase makes up about 90% of the cell's entire life. This is all the behind-the-scenes prep work where the cell is growing and making a perfect copy of its DNA. The M phase, which stands for mitosis, is that short, dramatic finale where all that preparation pays off, and the cell physically splits in 2.
So let's zoom in on that huge chunk of time, interphase. It's really easy to think of this as some kind of a resting phase. But you know what? Nothing could be further from the truth. This is an incredibly busy time for the cell, where it's getting everything just right for the big division.
Interphase itself is broken down into 3 distinct acts. We've got the G1 phase, the S phase, and the G2 phase, and each one has a totally critical job to do before the show can go on.
First up is G1, or growth 1. And yeah, that's pretty much exactly what happens. The cell physically gets bigger. Its cytoplasm, the jelly-like stuff inside, expands, and it's furiously making new organelles, its tiny little organs. It's doubling its supply of things like mitochondria. It's also cranking out all the proteins and RNA it's going to need for the next step. It's a beehive of metabolic activity, all focused on one goal, getting ready for what's next.
And now we move into the S phase, or synthesis phase. After all that growing and prepping in G1, the cell is finally ready to handle its most precious cargo, its genetic blueprint. The main event of the S phase is singular and absolutely monumental: DNA replication. The cell meticulously creates a perfect, error-free copy of its entire set of genes, making sure that both new daughter cells get an identical set of instructions to live and function.
Okay, this brings us to something that trips a lot of people up. How do we actually count chromosomes? The golden rule is really simple. You just count the centromeres. That's that little pinched-in part that connects things. The number of centromeres is always the number of chromosomes.
And this slide shows you exactly what that means. Before the S phase, you have 1 chromosome, just a single structure. Pretty simple. After the DNA is replicated, that chromosome now has a perfect copy attached to it, forming that classic X shape. These 2 identical halves are called sister chromatids. But here's the key. Even though we've doubled the amount of DNA, it's still considered 1 chromosome because it only has 1 centromere holding it all together.
After that huge job of copying all its DNA, the cell enters the G2, or growth 2 phase. You can think of this as the final paranoid proofreading step, right? The cell treats its genetic code like a top-secret document. Any tiny error in the copy could be a disaster for the new cells, so G2 is that final quality control check to fix any mistakes before it commits to dividing.
So, with all the preparations complete and the DNA checked and double-checked, the cell is finally ready for the main event, the M phase. This is where 1 cell dramatically becomes 2.
The M phase, or mitosis, is probably the part of the cycle you remember from high school biology, with stages like prophase, metaphase, anaphase, and telophase. It's the actual physical process of carefully separating those duplicated chromosomes and splitting up the cytoplasm to make sure 2 new, genetically identical daughter cells are formed.
So, we've seen all the steps of the cycle. But a process this precise doesn't just happen on its own. It's not random. And that begs the question, who or what is in control of all this?
I mean, really think about it. What tells a cell, okay, it's time to divide? And, just as importantly, what gives the signal to stop
to say, hold on, something's not right here. There has to be some kind of control system, right?
And there is. That control system is basically run by two opposing types of genes. You've got proto-oncogenes, which act like the gas pedal. They're the go signal pushing for cell growth and division. And then you have tumor suppressor genes, which are the brakes. They're the guardians that can slam the process to a halt if they detect any errors. For a cell to divide the right way, the gas has to work and the brakes have to be completely reliable.
And if we zoom in just one level deeper, the actual go signals are given by a pair of proteins. First, you've got cyclins. Their concentration levels go up and down in a predictable, well, cycle. Makes sense, right? They act like a key. They bind to and turn on these enzymes called cyclin-dependent kinases, or CDKs, which are like the ignition. So when the key, the cyclin, turns the ignition, the CDK, the engine starts, and the cell is pushed forward into the next phase of the cycle.
So when you look at the whole picture, the cell cycle is this incredibly elegant dance of go and stop signals, of a gas pedal and a brake. Which brings us to our final and most critical question. What happens when a cell's programming gets corrupted? What happens when a cell just forgets the steps to the dance?